This Week in Microbiology is brought to you by MicrobeTV and is supported by the American Society for Microbiology. To learn more about microbes, visit microbe.tv and asm.org.
This is TWiM, This Week in Microbiology, episode number 345, recorded on November 13th, 2025. I'm Michael Schmidt from the Medical University of South Carolina, and joining us today on this episode of TWiM, where we explore the unseen life on Earth, is our TWiM colleague from the University of Michigan, Dr. Michelle Swanson. Welcome, Michelle.
Hello, it's good to be with you.
Can you believe that IU is still undefeated in football?
Number 2 in the NC2A rankings. Yeah. And that's Indiana University, for those of you who aren't familiar with Big Ten sports.
And unfortunately, we don't get to play Michigan this year. We're going to finish out our regular season playing Wisconsin on this weekend. And the last game in the regular season is the old Oaken Bucket game at Purdue in West Lafayette, Indiana. Also joining us from Washington University is Dr. Petra Levin. Welcome, Petra.
It's great to be here. I am very happy to see you guys. And let's see, I don't really know much about Big Ten sports, although like my uncle would now be very upset with me since my entire family, at least a generation before me, went to Michigan. But let's say, I think I am from a D3 school, and sadly, my D3 school, Williams, lost to its rival Amherst this year by 1 point. So next year, Eves, next year.
So the question that our listeners want to know is, how many good jokes did the children of St. Louis offer during their Halloween adventures?
Oh, yes, that is a great question. We got some excellent jokes. Let's see. One of my favorites was, what is so great about Switzerland? I don't know, but its flag is a big plus.
Ooh.
What did one wall say to the other?
I don't know.
I'll meet you at the corner.
Ooh.
One of our trick-or-treaters had a notebook with her that she was writing down jokes in. So that was also quite lovely.
Wow.
Well, that's
like
a neat tradition that I had never heard about until our previous episode. If you haven't heard that one yet, it's about chocolate and other food items, namely ant yogurt.
Yes, and that has a bunch of excellent, at the very end, microbiology jokes. Mark had several longer, kind of more winding ones. But yes, excellent.
Let's
jokes.
Introduce Mark.
Yeah, I'm getting. And speaking of Mark, welcoming from the University of Puget Sound, Dr. Mark Martin, who is our TWiM adjacent colleague and host of Matters Microbial, a podcast that drops every Thursday. So welcome, Mark.
It's great to be here. I did not see any of the beautiful auroral displays because it was all cloudy and dark, but it was quite an interesting G4 storm, as my lovely mathematician bride reminds me.
And we even had some northern lights here in Charleston, South Carolina. It was a big storm.
And by the way, Petra, I can't believe that viruses and bacteria would just invade my body without permission. It makes me sick.
So, Mark, thank you again for pinch hitting, even though our baseball season is only just over. This week, our colleague Vincent is up north in Toronto at the American Society for Tropical Medicine, where he and Daniel are likely recording a podcast during that exciting meeting. If you like learning about the science of microbiology, please consider donating to MicrobeTV, a 501c3 not-for-profit organization where your donations made through November, December, and January will be matched by Parasites Without Borders to help us to continue bringing you this podcast. We greatly appreciate your support. I'm going to get us started this afternoon with a snippet from an open access paper that was published in the October 23rd issue of the journal Nature. And the paper is entitled Microbial Iron Oxide Respiration Coupled to Sulfide Oxidation. And the paper is authored by 13 individuals affiliated with 8 different departments at universities in China and Austria. And they are Chen, Li, Battisti, Guan, Montoya, Usvadik, Javek, Pollock, Richter, Schintelmeister, Wanek, Moosman, Loy, and it's an absolutely fascinating paper. I'm actually buzzing about this paper. It's a fundamental discovery that, I hate to say this, but it's going to literally rewrite the textbooks on global element cycling as it truly connects the deep earth to the global element cycles, namely the global elements being iron and sulfur and oxygen. And it brings a new acronym into our TWiM lexicon, namely MISO. No, this is not another food episode, nor is the paper about soup, but rather it's about microbial iron oxide respiration coupled to sulfide oxidation. This study in Nature fundamentally changes how we think about the sulfur in iron cycles. Now, don't worry, I'm not going to go into all the gory details. I'm going to make this approachable for you all. For decades, we thought that when toxic, stinky, rotten egg smelling sulfide met up with iron or rust, the iron 3 oxides, in the anaerobic muds of marine sediments, it was just a slow, boring, abiotic chemical reaction. But Chen and the team said, hold my multi-heme cytochromes, and they just blew up the model. They've proven that microbes, specifically microbial iron oxide respiration performers, these are the mesobugs, are actively coupling the two processes. Now, folks, this isn't just a lab curiosity. It's a massive, globally relevant process because iron oxide represents one of the largest pools of oxidants on the Earth's surface. The ability of microbes to step in and mediate this link between the sulfur and iron cycles means that they have a fundamental role in global biogeochemistry and perhaps even the planet's redox state over geological timescales. Now, this paper demonstrates that microbes are actively mediating this connection using a genetically sophisticated extracellular electron transfer system. The mesometabolism is thermodynamically favorable even in neutral and alkaline environments where typically iron 3 reduction stalls out. Once you got rust at neutral or alkaline environments, you still have rust. But the microbes have figured out how to turn that rust back. This is a massive update to our models of deep sea sediments, wetlands, and perhaps even aquifers. It's a foundational piece of microbiology that clarifies a long-standing biogeochemical mystery. And it's absolutely fantastic the way they go about demonstrating the science behind it. The sheer scale and depth of the study are a huge strength in this manuscript. They just didn't stop at a predictive genomic survey, which in itself was impressive because they actually looked at the metabolism across 37, that's right, 37 different prokaryotic phyla. This tells you this isn't a metabolic fluke of 1 or 2 microbes. It's a globally significant, widespread phenomenon that's observed in metabolism in nature. So just take that, E. coli and salmonella, when we get to you guys in the next paper. They also performed a classic, if you will, TWiM-style validation experiment using a cultivated microbe, De sulfovibrio alkaliphilus.
And this is actually awesome because they did the validation. Often you read these studies and they don't do the validation. So I want to point out, like, kudos to them.
And it wasn't a small piece of work. The figure goes on for a whole page, folks. And since it's open access, you can actually dig into each of the specific figures. And so what they did is disulfur alkylipholis can grow chemolithotrophically by coupling the sulfide oxidation. So they're going to use the sulfide to donate the electrons to the dissimilatory reduction of nitrate and nitrite to ammonium.
So can you break down, Michael, sorry to interrupt, but
I'm going to do that in a second.
Okay, but what chemolithotrophic means, because I know that Mark and I throw these out to our undergrads and they just we are chemo hetero.
We are heterotrophs.
Yeah.
Heterotrophs.
Tell us what that means, please.
Tell us what those words mean before you get into the details.
All right. Chemolithotroph means they're literally eating rocks, folks. They're consuming the inorganic material as their source of energy. And as Mark will likely interject, they have rubisco. So they're going to use the electrons from the hydrogen sulfide to actually fix CO2. And so that's what chemolithotrophy means, is it uses an energy source and they're burning they're burning not carbon, but hydrogen sulfide. They're stealing the electrons from the hydrogen sulfide and using then rubisco, they're putting that into carbon to make more disulfovibrio. And
rubisco is
ribulose 1,5-bisphosphate carboxylase.
The most abundant enzyme
on planet Earth.
Yep. We usually associate it with plants.
Yes.
Yeah.
So the important thing here is, and this is what I think certainly like new students in microbiology and anybody who's taken a biology course where you learn it from the eukaryotic animal mitochondrial and aerobic E. coli bias, is we use carbon to also make more of ourselves, but we need to eat organic carbon that is fixed. Carbon dioxide is not an organic, it is an inorganic. All other carbon, like if you eat trash, if you eat a hot dog, if you eat candy, that carbon we use to make more of us and animals that do that. And then we also use that carbon to generate reducing equivalents, so NADH, NADPH, which goes to the top of the electron transport chain and becomes NAD+, and that proton then goes down and gets pumped out to generate proton motive force to generate ATP. These guys are amazing. They live on air and rock. They use rock, basically, and Michael will tell you what, instead of NADH, so non-organic things at the top of the electron transport chain, and then to generate ATP. And they also use that energy to fix carbon dioxide to make more of themselves. So they are essentially completely independent of anything organic. They make the organic by fixing the carbon. Chemolithotrophic. And a lot of bacteria do this. And I think most plants also do this. So this is a pretty common thing, but I don't think we're taught the right way. So
we're not taught the full spectrum.
We're not taught the full spectrum, and we are so biased towards NADH and oxygen.
Right. And can I just, and I'll keep this very
smart,
but I just got through teaching this stuff. So I have to tell you, I have what I call the 3 deadly centrisms. And Michelle, the first is eukaryocentrism, because that's the way it's taught. When I was talking about sources of electrons and donors and recipients, and you have a table full of things that microbes use. Second is oxycentrism, more of the same. And the third, I would argue, is colicentrism. And this is a really good example of that. So once again, I mean, when we talk about rubisco, this is a bacterial enzyme. Sorry, friend, it is. All the really cool things eukaryotes do, they kind of stole.
Yeah.
Cyanobacteria gave it to chloroplasts.
It's true.
Became the chloroplast.
It is indeed. And if you are interested in things like this and you want to read a piece of popular fiction, there's a book out called Project Hail Mary by Andy Weir. that sort of touches on all of the things that Petra just went over in a fun way. But back to the paper. Using desulfovibrio alkylipholis, they actually demonstrated that this organism literally grows by breathing, underscore the word breathing, iron oxide, and then using that energy to turn toxic sulfide into harmless sulfate, which doesn't have an appreciable odor.
So in this analogy, the iron is compound is
oxygen. Yeah. By demonstrating that this microbe can grow autotrophically, where it fixes CO2 by taking sulfide, even iron monosulfide, FeS, and oxidizing it all the way up the electron transport chain to sulfate using solid extracellular ferrihydrite as its electron acceptor. Crucially, they showed that the biological processes outpace the abiotic chemical reactions at relevant concentrations, proving that this is an ecologically relevant mechanism. And the ability of this microbe to use what are essentially microbial nanowires, which are nothing more than the outer membrane cytochromes, to physically move electrons from the microbe or the bacterium to a solid extracellular rust is just phenomenal. Then, if that wasn't enough for you, the transcriptomics data that the authors offer as evidence behind their beautiful figures concerning the mechanism confirms that the mechanism involves the reversal of the dissimilatory sulfate reduction pathway to oxidize sulfide. As an aside, dissimilatory sulfate reduction is an anaerobic respiration process used by some microbes to get energy by not oxidizing sulfite, but by reducing sulfate, giving its waste electrons to this SO4, just like these microbes are giving their waste electrons to the rust, resulting in the sulfur being transformed back into sulfide. This dissimilatory part means hydrogen sulfide is then excreted as a waste product, and in the process, it generates energy, distinguishing it from the assimilatory process, where sulfate is used for biosynthesis to build cell components. And this process is crucial for global biogeochemical cycles and often occurs in very oxygen poor or anoxic environments. But here's the kicker. They found a very heavily upregulated multi-heme cytochrome that is homologous to the infamous OMP-CS that we remember from Geobacter, a gram-negative that was made famous by Derek Lovely and all his collaborators over the years. And they're the ones that actually coined the term microbial nanowires. Yes, folks, these microbes are channeling electrons to a solid mineral outside the cell using what look like tiny, tiny wires. Talk about infrastructure building. It's mind-bending. And if that weren't enough, here's the final mic drop bit. The biological microbial iron oxidation respiration, or MESO process, actually outpaces the abiotic chemical reaction at environmentally relevant sulfite concentration. Who knew the microbes were faster than the rocks? But there's always a but in TWiM. And so while the genomic predictions were incredibly broad, again, they identified over 5,561 species across 71 file with this potential, but they are currently uncultured. So the rigorous experimental validation that they did of their observation is largely focused on one of the pathways of the cultivated star, namely the desulfovibrio. This means that microbial investigators still have a colossal amount of work here to validate the mechanism and scale of those other two MESO processes that they well describe in all those other predicted organisms, and finally figure out the impact of the uncultured majority. However, it leaves the door wide open for future work, which is why we love this kind of paper. The other nit I would offer our listeners is that this, and this is not slighting the authors in any way, is that while the paper predicted 3 metabolic options, the robust experimental validation was only done with the desulfovirial. We still need to confirm the other 2 pathways that are predicted from the metabolism. Still, the novelty is off the charts. I thought that iron and sulfur cycling were connected primarily through this passive chemical precipitation. And this paper again demonstrates what Mark said earlier, that microbes will continue to amaze us and that the microbes are actively mediating this connection using a genetically sophisticated extracellular electron transfer system. Mesometabolism is indeed thermodynamically favorable, even in these neutral and alkaline environments where typically rust is made and iron reduction stalls out. This is going to serve as a massive update to our models of deep sea sediments, wetlands, and aquifers. It's truly going to become a foundational piece of this century's microbiology and clarifies a long-standing biogeochemical mystery. This is absolutely fantastic, and it likely has the scientists at the UN Climate Change Conference in Belém, Brazil, buzzing. So I encourage you to take a look at this paper, and I'm going to turn this over now to Petra, who's going to take us back to E. coli and Salmonella. Petra?
Yeah, I'm going to take you back to E. coli and Salmonella. So this is actually a paper that came out this week. So it's truly this week in microbiology. So this paper, I actually was at a meeting, and I'm hoping maybe we can talk about some of the other things I heard at this meeting that were microbial. It was a meeting on the sort of connecting cell biology to evolution.
Ooh.
And I will get to this paper, but I also learned about teeny tiny fossils that are shaped like little mini vases, and you can see almost little animals in the little fossil vases. They're 10 microns tall, and they essentially probably have some organic left on them, so they're hoping they can kind of identify them. I learned about the evolution of chloroplasts, fascinating stuff, evolution of respiratory chains in mitochondria and in the alpha proteos, who are sort of the ancestors of the mitochondria. I mean, there was a lot of amazing stuff.
Do you have a great job or what?
We have we are so privileged. Yes. But this paper is about a kind of motility, which is almost a passive motility, I would say. And the title is Swashing, a propulsion independent form of bacterial surface migration. It's by Dustin Panich, Eric Dudabout, David Blair, and Navish Wadhwa. And they're all have been affiliated at some point with the Arizona State University, which is actually where the meeting was. And I really enjoyed this talk. The PI, Navesh, gave a really great talk on this. And it just was fun to also think about how bacteria move. And so any of the people who are listening have probably heard about flagellar motility, and usually E. coli or Salmonella. They're very similar. I think they're close. They're 99% identical bacteria. They're members of the gamma proteobacteria. They're diderms. E. coli is sort of the sort of commonest, Mark mentioned E. coli-centric, commonest organism we use in the lab. Even people who don't work in bacteria often use E. coli for cloning purposes or other molecular biology. But E. coli have flagella, and perhaps we can post a picture of E. coli and Salmonella. Their flagella structure is very similar. And they use these flagella for chemotaxis, is sort of how we're taught originally. And chemotaxis is if they spin their flagella counterclockwise, they have smooth swimming. But if they rotate their flagella clockwise, they tumble, and that's how they change direction. E. coli and Salmonella. Other bacteria do it differently, but that's how E. coli and Salmonella do it.
And the term chemotaxis refers to the fact that they can swim towards something desirable or away from something harmful.
Exactly. So if there's an attractant or a repellent, they alter their swimming and their running, what we call running, swimming, and tumbling, so that they bias it towards swimming away. So they might, if they'll constantly sample, they say, ugh, yuck, there's more of this repellent, and then they'll sort of change direction and swim, and they just are constantly sampling so they can move kind of away, down a gradient of repellent, or up a gradient of attractant, like a sugar.
And remember that both of these microbes are paratrichously flagellated,
Exactly.
which means they have flagella all over their bodies. And when the flagella get together, it's akin to effectively combing your hair into a ponytail. And so what they're doing is they're spinning their ponytail either clockwise or counterclockwise in order to facilitate the motility.
And kids, don't try this at home.
Yeah, don't try this at home. But it's the important thing to know about chemotaxis is it's an individual bacterial behavior.
Yes.
And so individual bacteria have these receptors, and they're constantly sampling, which up a gradient, down a gradient, and then changing whether they keep swimming or they keep running or they tumble. And this is done and the only reason I'm telling you is so that people understand, like, you can see this if you have 0.3% agar, so very low agar, and they can actually swim through the agar. So you can see this, and they will move out on a petri dish kind of to form like an ever expanding circle till they meet the edges of the plate. Okay, so that's one kind. That's the kind we all learn about. There's some beautiful genetics, super fun. The kind that sometimes we learn about or we hear about more is called swarming. That's on a harder agar, 0.5%. It's a collective behavior. In bacteria like E. coli, Salmonella, or Bacillus, which is also paratrichous, but a gram-positive Bacillota, monoderm, they often become hyperflagellated when they swarm. It's much faster, actually, than chemotaxis. And it's they become also biased towards running, so little tumbling. And again, they move out in an edge. So it's not quite swimming, but it's not swimming. And these guys can be helped, actually, if you get rid of some of the friction in the plate by adding something called surfactant, which is just soap. So, if you put down soap, it becomes slippery. So you can help things swarm that way. Some bacteria, Myxococcus and some other ones that don't use flagella in the same way, they actually put down surfactant and kind of just glide. And that is also called swarming, and it is also a collective behavior because they do it at a group. If you look at the front of a swarm of a plate where they're swarming, you can see them kind of all together, kind of moving more, at least the ones in the front are moving pretty quickly forward.
They're making their own slip and slide.
They make their own slip and slide with the surfactant.
And
but again,
we apologize for the earworm that now everyone has, seeing Paul Simon slip sliding away.
Yes. But I think the important part about swarming is it's still a flagellated response, at least in E. coli and Salmonella. Okay, so here, at least two of the authors on the paper, they were looking at Salmonella, and I think they were doing some controls. And we'll upload a picture of a flagella, but essentially for the purpose of this discussion, the flagella have a motor. It's really cool. The structure was solved maybe 25 years ago. The motor is on the inside. It's sort of in the inner membrane, right? These are diderms, kind of stuck in the inner membrane. And then they have parts of it. There's a stator and different parts. And then as you get to the outer membrane, you have what's called a hook, which is sort of like the lower part of the filament. And then you have the longer part of the filament. So the flagella itself, sort of the part that actually gets turned and generates propulsion, has two parts, the hook and the filament. And then on the inside, you have the motor. And then you have some things that regulate which direction it turns and the force and things like that. So what they noticed in Salmonella is that if on this specific media, so 0.5% agar, top agar, they could get motility. The cells still appeared to swarm, move out, even when they had mutations in the hook or in the filament.
And I appreciated that they described this as an intended negative control.
Exactly.
And yet when they looked at the plates, they could not deny that there had been migration.
Exactly.
So,
and it's not slow. It's about half the speed.
Was it a contaminant? What's going on?
Exactly. So then they start making mutations, and they find that if they make a mutation in the motor so that the flagella can't turn, they actually don't move. So they have a protein called mode A. If they get rid of that protein with a mutation, the cells don't move. But then they did one more experiment, because you might think, well, maybe the motor's turning whatever remnant there is of the flagella. They actually got, so moat mutant, you still have the filament out there. So when they get rid of the filament and the motor, now they move again. And that, they say, is probably that the flagella that are paralyzed because there's no motor are maybe getting in the way. I mean, they're long, there are a lot of them.
Kind of a drag on the
They're
movement.
dragging. Who knows?
And remember that flagella spin at 100 hertz, which is 6,000 RPM, and they consume about 1,200 protons per revolution. So this is not an energy non-intensive process. It costs them some ATP.
Yes, but I want to point out, once you delete the motor, nothing's happening.
That's right.
ATP. And these guys are also growing under conditions where they have more ATP than they need. They're growing on LB. I think they're growing on sort of a rich undefined media with extra glucose, which we'll come back to because that's important.
You know, I wanted to say something, Petra, that Bill Metcalf, University of Illinois, Urbana-Champaign, used to tell me that a phenotypic screen is for your benefit, not the microbes. They have a different agenda. And this is a great example of that, because I like it when I think it's going to be something and it isn't. That's cool. But I have seen scientists get really frustrated.
Yeah. And I think actually what I was told is you get what you screen for, not actually what you want. I guess that's the other way of saying it, because we do a lot of genetics. And so, they, but again, this was their control. It wasn't even, they weren't even looking for it. Okay, so that's really cool. And then they're trying to figure out kind of what's needed. So they go to E. coli, which again is very similar. They actually do something I thought that was cool in salmonella. And this is kind of one of these big open questions. They then delete, so there's a sigma factor that turns on essentially the genes to make the flagella. When they make a mutation in the sigma factor needed to make the flagella, they don't move. So that's really bizarre, because if you delete the motor and the flagella, what's the difference if you delete the thing that drives the genes that make those proteins? And then if you delete the thing that inhibits that transcription factor, you also don't move. So that's a little mystery, like why, when you delete the things that turn on and off synthesis of the flagella, do you no longer move? So that's a little bit of the mystery, unclear, but another paper, something else. They then move into E. coli, and they're trying to understand what's going on, because this is really not expected. And they think about, mentioned bacteria that can swarm that don't have flagella. And those work by putting down surfactant. And so they sort of have a little slide. So like the bacteria kind of slide forward on the sort of soapy surfactant. And that's what's allowing them to move. And so they're wondering maybe if that's possibly involved. And so they put down a couple kind of soaps to see, and actually at higher concentrations, they interfere with this motility, which they are calling swashing.
So again, another observation that's contrary to what they were anticipating,
exactly.
Which is that's why we call it research and not search.
Exactly. I mean, I love this paper because it is a journey, this paper, and the talk was a journey, a fun journey. So they're testing the one thing that they are aware of, that unflagellated things allowed to move, surfactant, and it was getting in the way. So they have to kind of take a step back here. And there's no growth problem, because you could imagine like the cells are just growing and pushing out, and none of the things that they've done have altered the growth. So it's not just growth. Okay. So we talked a little bit, Michael talked a little bit about sort of this chemolithotrophic metabolism. E. coli are organoheterotrophs. They're organoheterotrophs. They use, like we do, fixed carbon for growing, and they use sort of NADH that they make as they go through the glycolysis or TCA cycle. they can do two things. They can take that NADH and they can put it into the electron transport chain and make, reduce oxygen, generate proton motive force. E. coli and Salmonella, though, even when they're growing aerobically, they do something we call mixed acid fermentation or overflow metabolism. And it's still a little bit of a mystery, but the best explanation I've heard for overflow metabolism, where instead of essentially taking acetate and then putting it through the citric acid cycle to make a whole bunch of vitamins and cofactors and things like that, and more NADH for the electron transport chain, they instead just take that acetate and they dump it out. So it's a 3-carbon acidic molecule. They just dump it. Because they're growing here on media, they don't need to run the TCA cycle. The TCA cycle is slow. They need ATP, but they can generate enough just by whatever bits of the electron transport chain they're doing, and then also substrate level phosphorylation, where they just generate ATP as they go through glycolysis. So at the same time they're running aerobic respiration, they're also doing a kind of fermentation. E. coli and Salmonella. E. coli mostly I think pyruvate acetate, and I think Salmonella dumps acetate fumarate is the combo. But anyways, they're dumping these acidic molecules. If they feed the cells something that doesn't really go through glycolysis the same way, so they don't really have these products to dump, they're not spitting out acidic carbon molecules, they don't swash. Mm-hmm. Okay, so
there's a clue.
That's
a good example is glycerol.
Glycerol. Yeah, they are actually giving them, I think, I wrote down what they were giving them, but some other things that don't get dumped like this. Salmonid formate and acetate are Salmonella, and E. coli is mostly, I believe, pyruvate, maybe a little lactate. So in any case, they're dumping acidic molecules. The ones at the front, right, of the swarm have the most to eat because they are growing on fresh media, basically.
They're not competing.
They're not competing like the guys behind them.
Right.
So they actually do a little pH test. They pH the medium, and they find, not surprisingly, that the medium is about 6.86, which is about what you're pHing LB to when you make it. The front, right where the bacteria are, as much as they can get resolution, are pH 6.1, so almost an order of magnitude lower pH. And the center, I wrote down, is 5.35, so quite acidic in the center of the colony where they originally did. So there's a big gradient because, again, each pH value is a full order of magnitude from the center to the edge of the plate. So that's another clue, but also you have a lot of molecules they've dumped to get that to happen. That drop in pH is all these acidic little carbon molecules the cells are dumping as they're just gobbling as fast as they can, the carbon and glucose from the LB glucose.
And what's interesting about this is we usually talk about like doing single colony purification as though each colony is pure, it's identical. But in fact, there's a lot of really interesting metabolism and competition happening, and waste products that you're going to describe the implications of.
Exactly. Right. So even in colonies, right, even though maybe they're mostly genetically identical, and we could discuss that forever, but each bacteria is an individual, and we forget that. We treat them like they're all the same, some kind of average idealized organism, but that doesn't work any better really for bacteria than it works for people.
Yeah, some are in a much more dense environment. They're competing with a lot more cells.
Much tinier, denser, but individuals. But again, this is a collective behavior, so it is them all interacting. They actually show with the Salmonella, if they have a moat mutant that's kind of paralyzed, if they dilute it out with a mutant that doesn't make the filament, the moat mutant actually, even though it kind of usually gets stuck, gets dragged along. So this is a collective behavior. So they sort of speculate that somehow the drop in pH and again, pH, right, and all these molecules, you're changing the osmolarity, right? They're dumping a bunch of things, they're increasing osmolarity where they're sitting, and then it's lower farther out. And then actually figure 4 is amazing because they look at the colony topography. And so they do it in a couple different ways, but the figure E, they actually have the model, because what they think is happening is that the edges, so the topography is it's sort of like a flat plateau, and then there's a little bump, and then it goes down. So the edge of the colony is almost like the ridge of a plate, but then the plate ridge goes way down. So actually it does look kind of like a plate. So you can imagine that ridge. And so they have this really nice picture or model in figure 4E, where they have this bulge of fluid, because when you have high osmolarity, right, water comes in through diffusion to dilute that out. So you have almost the bacteria riding a wave at the front and like surfing on it, or
They're
swashing. And then they kind of get pushed out as they do. So the bacteria are making their own osmotic gradient, and that's driving them forward. It's really cool. They call it swashing, and they liken it to what happens when a wave comes in on an ocean up into the beach, and you get that very thin film, and there's kind of organic matter. I love to look for beach glass. My daughter and I are kind of obsessed with doing this at the beach, but that's a good place to look for beach glass because it's shiny, but also gets pushed by the fresh waves, that kind of wet area. So that's how they kind of see it. So the bacteria are kind of creating their own wave by secreting all this acetate and formate and sort of molecules, and that's pushing them forward.
And pulling the water out of the agar.
Exactly. And
naturally
pulling the
flows.
water out of the agar. They're sort of making their own wave, and they call it swashing. And I think this is really cool. It's kind of a fun thing to read about, but it's also, I think, brings up something which maybe goes back to this idea of that the bacteria have their own agenda. But here, I think there are a lot of phenomena we look at in bacteria. My lab studies size, and one of the questions we get asked a lot is whether size is actually why the bacteria have this size, why they are the size they are. And we can come up with all sorts of reasons, but I think in this case, and maybe some in size and some other things, like why do they have 1,000 flagella instead of 500, right? Some of this is just kind of how evolution ended up. This is a physicochemical process here. It's, are the bacteria doing this on purpose? I mean, obviously the bacteria are not individually thinking, I need to do this. This is just the way their metabolism works in the lab. But whether this actually is something that has been adaptive, I think is really an open question. It could just be in this very specific lab situation where we're growing them under very specific 0.5% versus 0.3% agar, fermentable sugar like glucose. It happens. But I think this is really important because sometimes why we always say is the hardest question to answer in biology, but I think sometimes the answer might just be physics and chemistry. And I don't know for sure that's in this case, but they haven't really, I think the authors themselves are really agnostic about it, and you could imagine it being to the advantage of the bacteria in some way. They get to move out, even if they don't have flagella. But you could also imagine that it's just a consequence of their metabolism plus the conditions that they're given on the plates that lead to this. And I thought that was kind of fun to think about. And I don't know. It's just a really nice paper.
But the other thing that I think that's driving this fitness is at the end of the day, you do not burn any carbon unless you have an oxidized, an electron acceptor. And in the case of E. coli and Salmonella, that electron acceptor, that initial electron acceptor for the burn is oxidized NAD. And it accepts the electrons. If you don't have enough oxidized NAD around, you can't do anything. You could be sitting in a vat of glucose, and if you don't have any oxidized NAD, there's no go.
Right. And I think that's the really big question about mixed acid fermentation or overflow metabolism, because when they dump the acetate, they're not recycling the way that they would the NADH. So normally in fermentation, when you get rid of the oxygen, you force them to ferment. And when you ferment, like if you make lactate or when yeast make alcohol, ethanol, that takes essentially takes the NADH and makes it into NAD+. And then the NAD+ can be used for more glycolysis. If you're doing aerobic respiration or anaerobic respiration with NADH at the top, that is where you recycle your reducing equivalents. And so it's always been kind of confusing why have this, essentially aerobic, not even productive fermentation where you dump acetate. And I think one of the things to think about the electron transport chain that I think is super fun way to think about it is that, yes, it's making ATP. And again, this goes back to the bias of how we teach these things. Yes, the electron transport makes ATP, but in this case, for the bacteria, they can make plenty of ATP, so much they can dump their carbon as acetate. But maybe the aerobic respiration is a more efficient way to recycle the NADH into NAD+, so they can keep gobbling up the sugar.
And the other thing that both Salmonella and E. coli have is they have an enzyme called formate hydrogen lyase. And so that's the ultimate waste product because remember, hydrogen gas escapes Earth's gravity. And so consequently, if your waste product is hydrogen, off it goes, and other microbes in the community, because again, pure culture is not the way of the world. Hydrogen is a wonderful energy source for other microbes. And it could be this dynamic between the formate hydrogen lyase and substrate availability. And that's the challenge with mixed acid fermentation, is because you don't know which way the pathway is going to go. And it goes based on the needs of that individual and not the community. Yet we study communities.
Exactly.
I love this paper so much. But, I wanted to say something about it. Every time that I look at a colony, I think about this because I study I shouldn't say I study, I'm not that cool. But I've noticed I see variation due to stressors that take place within an aged colony. And I've been doing that for many, many years. And it's an interesting idea. So when you hear about the way that a colony appears and the way that it negotiates living on a plate, look at Panabacillus, for example, which is almost artistic. You think to yourself, is that relevant to nature? And when I say that, it's not a judgment, and people sometimes hear it that way.
Tell us what Panabacillus looks like in a colony.
You get beautiful, like pinwheel appearances of that, and there's some evidence that they actually help to carry spores of other organisms along with them. So Bar Eshel the late Bar Eshel did some wonderful photography of these things. They're very artistic. If you do a Google search on Panabacillus, you will get these beautiful images. You ask yourself, is that relevant to natural history? And it may be an emergent property of some elements that are taking place. So when I heard about this swashing business, first off, I went to swashbuckling because I thought that was pretty funny in my mind. Well, it is. It's a swashbuckling story because it's buckling the auger. But what is interesting to think about here is, does that relevant to other places in nature that they might be? And it is, because you're pulling water out of some area, you're causing a physical deformation. And by the way, I love the idea that they're surfing, because I've been looking for surfing bacteria for some time. So here you've got a really good example of it. And what I want people who are very coli-centric to do is to think about other environments in which this might be relevant, because I don't think this is just about a colony at 0.5%, is what I'm trying to say.
Yeah.
I think it's deeper than that.
Sure. Yeah, because these are basic principles, right? Mm-hmm. Water flowing down the concentration gradient.
And this, kids, is why we take physics.
It comes back to physics, Michelle, and something that I'm not super strong in. But if you think about it, and they actually mention this in several places, especially with regard to surfactants, we don't think about that. And I was accused more than once of thinking intentionality applies to bacteria and archaea. Guilty. But what I am saying with that is we tend to ignore the physical nature of what we're looking at. For example, on Petri dishes, there is a thin layer of liquid water on top. Mm-hmm. And we tend not to think about that very much.
Yeah, I would like to point out this lab is in the physics department.
Yeah.
So, well, I mean, they're in the physics department. And I think, I was just joking around, but like as biologists, we take physics and we take chemistry, a lot of chemistry. And, the organic chemistry is kind of the core of molecular biology. But, the physics and the inorganic chem, in a way, that's what's at play here. And
that's the perfect setup for me describing the biography of one of the first authors, Eric Durabout. He grew up in a family of engineers, so he was just surrounded by math and physics. but he was very curious about biology. He found that as an undergraduate at Arizona State University, he didn't have a course that really brought the physics and the biology together. But then he learned about Professor Navish Wadhwa joining the faculty and was exploring the intersection of physics and microbiology. So he thought, aha, I would love to join this. And he was fortunate to join as an undergraduate researcher and did some work then. He's currently now a first-year medical student at University of Arizona and did this work as part of his research experience. So he got some experience as an undergrad in an informatics and computation and open neuroscience lab. He also was able to get funding from the National Science Foundation Research Experience for undergraduates, which allowed him to do some bioinformatic type work, and studied circadian rhythm movement and health questionnaires from people. It was part of the analysis that he did as that summer student. And then he's been in the Wadhwa lab for a couple of years now, and wants to give some credit to people that influenced him early on. He said that he had no wet lab experience when he joined the lab, and his fellow lab mates, Luis Meninas, Carolina Gogarty, and Shabduli Sawant, took time out of their own busy schedules to train him from the ground up, and that made this work on bacterial surface motility possible. And then he also really appreciated when Professor Blair reached out to the Wadhwa group to describe this finding and ask for a collaboration. In particular, Ren Osar was investigating it with Eric through a computational fluid dynamics modeling system. So although the manuscript is really focusing on the experimental side of the swashing, he credits Ren with her ideas, taking it to really think deeply about the understanding of the behavior. He also said what really influenced him and motivated him during this swashing project was his curiosity about the behavior, and also just the great support he got from others in the group. And he said finding a research project that's both rewarding and allows you to work with kind people, collaborative people, makes science so much more enjoyable. And speaking of enjoyable, he enjoys hiking with his family, great way to relieve stress and collect his thoughts. And he's so happy that Phoenix, where he's been since an undergraduate, has a lot of natural beauty and good weather. So it's great to add balance to his work as a first-year medical student and a researcher.
Well, thank you, Michelle and Petra and Mark. This concludes another episode of TWiM. If you like learning about the science of microbiology, please consider donating to MicrobeTV. We greatly appreciate your support. We would also welcome your questions and comments that you can contact us directly at the email twim@microbe.tv. Michelle Swanson is at the University of Michigan. Thank you, Michelle.
My pleasure. Thank you all.
Petra Levin is at the Washington University in St. Louis, where she likely will be swashing her way through her laboratory, inspiring her team.
Thank you.
And Mark Martin is at the University of Puget Sound, where we know he continuously inspires his students in the lab. So thank you, Mark.
That's kind of you to say. I do my best.
And I'm Michael Schmidt, and you can find me here at the Medical University of South Carolina in Charleston. I'd like to thank ASM for their support of TWiM and Ronald Jenkins for our music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone, and see you next time on This Week in Microbiology.